All right, thank you, Lorenzo, for sharing the fix to the CompSol player. I think it's a good time for us to start this new module. Please remember that this lecture here is being live streamed and recorded on YouTube, okay? And so today and for this next hour, I would like to talk to you about the mechanisms of the Cellular Potts or GGA model, how it works, okay, how things are implemented, what makes the cells move inside the system, and what you can do to change, and what's going on behind the the screen, so to say. So it's going to be module 1.3 Cellular Potts intro. These are not acknowledging about the funding source for the CompSol 3D project. Okay. If anyone has any issues with the audio or the video of the slides, let me know. Okay. I guess these slides are going to be made available to you on the Google shared folder. If not now, very soon. So remember that some Zoom logistics for this uh, well, your screen sharing and microphones be disabled. You can use the buttons to go ask me to answer questions or to ask me to go slower or goes faster. You can add your questions or comments in the Zoom. There will be someone monitoring the Zoom chat, okay? And anything that's very urgent, they will let me know. And I guess you have been seeing this, and the views and PowerPoints are made available to you to review the material if you need, okay? All right, so reminder, these are tentative course outline. Yesterday it was the day 0 where you just introduced Python for those that not familiar. Now we are day 1 and you are in module 1.3, concept to the basics and intro to Cellular Potts modeling. And you still have one session over this one, and this is going to be the schedule for the remaining of the week. So, and then also day 4, 5, 6, and 7. All right. So this is going to be the topics of this module. Is only 2 topics. What first cell modeling, and then a brief intro to CompCell 3D, Cellular Potts Model, GGH basics. Basically, that's it. And I like to start by showing a picture of what you are trying to model in CompCell. So usually when you use CompCell 3D, when you make a model in CompCell 3D, what you're trying to model is our cells, okay? Cells and tissues or single cells, okay? So this is what you're trying to model, okay? This is what you're trying to understand using computer simulations. Now, how you can, so if you want to model this, well, you have to present this in the computer. And when it comes to represent this on a computer, there are a bunch of options, right? So there are multiple ways that you can do this. One of them is when going more or less historically, is using what's called the cellular automata type of models, where you basically, well, your cells are in discrete space in space. So let's just use a grid, and every grid that's filled represents a cell that's occupying that particular space. Okay, so here you just overlay a grid on your system of interest, and whenever there is a cell nuclei, you fill this grid with some number that represents that there is a cell there, and maybe the number represents the cell type. And then what you have here is that this representation for tissue, you have like 7 cells here, and this is the spatial distribution of those cells. So that is a way to present your model using cellular automata. Okay, you can also, of course, then assume that the cells have kind of discrete shape that is a square. Of course, this is just a model, a rough representation that was widely used many years ago when the computer power was not enough to do anything better than this. Okay. The other way is you can actually represent your cells as a particle. Okay. So, and then it's a lattice-free representation. Then the cells are radially symmetric, and they interact with one another by some kind of repulsive and active force, some usually a Lennard-Jones type of interaction, where there is a preferred distance that the cells would like to have with the other cells, or the particle would like to have other particle. There's a repulsive core, so they cannot get closer to that. And this interaction has a cutoff, so after this ideal distance, they can kind of push the cells together, but after, if they're too far away, they cannot feel each other, and there's no interaction with the cells. So that's the approach that things like PhysiCell use to represent cells in their simulations. Okay. Another, moving out to a little bit more maybe better approach, you can say, that take into account the cell shape. One of them is the vertex model, where instead of model cells are pointing a grid or a particle, you actually model, you still use particles, but the particles will be represent each vertex of the cell. So here you have a vertex here, 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 and then you connect them with some kind of line that has some kind of tension that provides a kind of cortical contractility of the cell. And of course, this model assumes that all the cells are confluent, they're actually touching each other. There's no free cell boundary on this kind of vertex models, right? Or maybe there are, but then it's a straight line, the cell has no curvature. But this allows some kind of representation of the cell shape. So it's a little bit better model in terms of translating what you see in the screen to what you have on your computer. Okay, and so these are three main classes. There are other slightly variations, a combination between those two that you can have. But for the CompCell 3D models, we use neither of these. What we use is we actually overlay a finer grid on top of the cells, and then whenever the cell is on it, we fill this grid with some kind of a number, and this allows a very precise precision of the cell shape, and the boundaries between the cells. I see that there is a question, someone raised it.
Yeah, so I had a question. So in the vortex representation, so you have two nodes connected, but there is kind of like a spring or a resistance in between. What does that mean? Or physical represent?
Well, that will be depend on the model. Okay, some models will not have a spring between them. It's gonna just be you just move the vertices according to a constraining area. Okay, but some of them they actually do have a spring. The springs can be either contractile or expansile, right? Can be under tension or compression. And usually in the case that it's under tension, right? You want to bring the vertices together, that can represent actin contraction on the cortex of the cell.
Okay, so the individual cells can change shape, right? Or their boundaries
Yes,
in real. Okay, great, awesome, thanks.
right. Okay, so and so it's a little bit better preserved cell shape, but that is also achieved by using the Cellular Potts model that we use in CompCell, because what we see here in the screen is very high resolution, but if you zoom in, is basically in any image that you take from a microscope is digitalized, right? So what you're actually seeing is pixels on a screen. So that's exactly the same approach that the Cellular Potts model used to represent cells on the screen. You just have a little bit coarser mesh, but you're basically the cells represented by occupied lattice sites on a regular grid. Okay, so hope that makes sense. Okay, so this is how you represent the cells on a screen. So that is not yet a model. To have a working model, you have to move those cells that you represent. Okay, so how do you move the cells in a computer, right? Or in your model. So in the case of the cellular automata, you basically are flipping a whole pixel to another one. So if the cells, this cell here is moving, the way that it moves is that you delete it from this pixel grid and copy it over to this pixel grid. Okay, and now the cell has moved. And a cellular automata, that's basically everything that cells can do. They maybe can change type, but if they move, they have to actually locate from one pixel to another one, right? In the particle models, then it's very easy, intuitive how the cells move, how they update their location. You just move from one center to another center, and that's based on energy minimization or force gradients, and then from that you sum up, integrate all the force there are around your particle, and you choose what's going to be the direction of the net force, and that's where you move your cell. Okay, or maybe your cell is self-propelled, so we already want to move in one direction and just move in that direction. Okay, and how much it moves will depend on its mobility, the amount of forces you're experiencing, the viscosity of the medium. There are things that can be embedded in this model. All right. The same thing is done when you doing vertex model. You compute the sum of all the forces in each one of the vertices, and then you move the vertex according to those forces, or according to kind of algorithm that minimize the energy. So we compute the local energy gradient around each one of the vertices and find the slope where you're going to minimize the energy, and then you move the vertices in that direction. Okay, and that's how you change the cell shape and eventually move the cell from one direction to the other. Okay, in the Cellular Potts model is also the same thing, but you are doing not remove the vertex of the cells, but you're moving the whole boundary of the cell. So not only this, but this and this and this. Okay. So the way it is done is by actually a little bit of the cell autonomy. You're flipping pixels, okay, around the boundary of the cell. And when you do that, you change the shape of the cell, slowly evolving it, right? And eventually you move the cell from one point to another, okay? So let's see how it works in practice. So here you have your simulation. So that's the initial layout of the cell. So let's move just one cell here. And now the way that cell moves is just by a series of lattice sites copy attempts, right? And then by flipping those pixels around the cell edge, you've changed the shape of the cell and can move from one point space to another point space, as you see here in this video, okay? So what's different between the cellular autonomy is that you're not only moving a pixel, you can actually make transient expansions and contractions of your cell, so you can actually deviate from this current volume, and you do that also based on some kind of local rule that decide where the cell should grow, where should shrink, where should move to, right? Okay, so how this is done. Now let's look at zoom in into this process to understand exactly how you flip those lattice sites, how you decide when you're gonna flip in one direction versus not flipping that direction. Okay. So a site copy attempt in the composite 3D models is done the following way. So first, it is intrinsically stochastic, okay, but it has to follow certain rules. So here on the left is the current state of the system, so that's your cell shape. And the way you evolve it is by first, you randomly select a site in your lattice. It can be any site, but if you select a site that actually lies near or at or very close to the boundary, okay, that is a site that can actually grow into another one. So you select the one on site I, and then you select this red one here. And after you select this one, you should select another site, will be a neighboring site to this one. So in this case, you select this green site right to the right of it. So that's your original site I, and then select another site J. And then you see if this J site is occupied by something else than the cell, maybe occupied by another cell, maybe it's occupied just by nothing, another object or the medium, then you can try to copy whatever is here into this. So that's going to be a site copy attempt that happens in the Composite 3D. So you may or may not make this copy depending on whatever you are following the properties or the behaviors that you encode in the cell. For example, the cell right now has this kind of volume given by the number of pixel sites that you occupied. And if you do this flip, this copy attempt, you'll be increasing the volume of the cell, okay? So that happens both in 2D or 3D, right? So in 3D, instead of having a plain lattice, you have a three-dimensional lattice, but the process is exactly the same. You select a voxel, right? And then select a neighboring voxel and you try to make the copy. Exactly the same process, okay? So, yes, you lead to a slightly shrinkage or expansion. So if this uh, pixel copy attempt drives the cell towards its target properties. For example, the cell wants to grow, okay? So it's in some stage of its cell cycle that it wants to grow for in order to divide eventually. Then this is actually something that actually will drive the cell towards its target properties. So in this case, you accept this copy, and then for the next frame, your cell is slightly bigger than before. However, suppose that the cells want to shrink, right? And in this copy attempt is going to drive the cell to grow. Then you can accept or reject with some probability. Do not automatically reject the copy attempt. You can still accept it, but there's going to be some kind of probability depending on how far you're moving your cell away from its target behavior. Okay. I see there's another hand
Yeah,
raised.
so I have a question. So you first select the random site I, and those random sites that you select in the first place are always on the boundary. Is that true? And never in the interior, right?
No, no, you select everywhere,
Everywhere.
anywhere. Yeah, everywhere, anywhere. Okay. And then you
So
select the
for
neighbor, anywhere. If those two neighbors are different, then you
uh
do the calculation. If you select, for example, the pixel on the left with the same cell, then they're right the same. There's no, then you just discard that whole process and then you start over.
so for example, if I select the cell in the middle, right?
Mhm.
So I don't like how can it migrate to any neighboring cells because there are already cells over there.
Yeah, well, you just select a neighbor, and because in the middle you will fail because you cannot find a, a peak site around with different properties. So you just do that, right? Let it fail and then try again. So this is done many, 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 many times. Okay, very
Oh,
fast.
okay, nice.
So there is no need to
Okay.
worry about selecting only the ones that are at the boundary. If you try to do that, your simulation is going to be slow because that's a very have computational thing that you have to do. So yeah, if you just randomly select, it's going to be actually be faster.
Oh,
Does that
nice.
answer the question?
Yeah, yeah. Thank you.
Okay. Hi then.
Yeah, I also just wanted to clarify, just to indicate more what we're looking at here. This blue area is one large cell. This is not a group of cells, and what's being randomly selected is one pixel within that cell to try to kind of reach out and swap. So it's not really the case that you would ever have a cell just completely switch into another cell unless your cells were very, very small and your temperature was high.
Mhm. Yeah.
That's all.
There's a comment from James that in another simulator software, they do have a keep of all the cell boundary voxels. The dynamics end up being the same. Um, I'm not sure if it's really um, good in terms of computation point of view if to make simulations low or not. Okay. Okay, but that's a detail. You don't have to worry about this. So that's more a question for computer scientists. You are modelers, and what you need to know is how it works. Okay. Okay, so now let's do another thing. So let's assume that this was accepted because your cell wants to grow, okay? And now what you have now is a new state where here's your cell and here's this new uh, occupied lattice site. So the cell is slightly bigger now. Now what you do another random selection. And let's say that this time, instead of selecting a site that is inside the cell, you select a site that's outside the cell. You select this site here, I. And then you do the process, you do a second selection of the pixels, the sites around this one. And let's say you select this one here. So what's gonna do now is that the medium or the void will grow over a site occupied by the cell. Okay. So this is a site copy attempt that actually is going to shrink the volume of the cell. Okay. So will you accept this copy or not? Again, that always depends on whenever this is driving your cell into its target behavior or properties or moving away from target behavior properties. So let's say that the cell is now in this size, and now it wants to shrink. Okay, if the cell wants to shrink, this is going to be a modification on the cell properties that drive it towards this property. So then you accept the copy and the cell shrinks. But or let's say that the cell is not done growing, you want to grow even more. Okay, so this will going to be a copy attempt that actually drive the cell away from its target behavior or its target property. So then again, you accept this with some kind of probability. You may accept or may not. You just have to flip the coin and see if you're gonna do this or not. Okay, so basically this is how it works. So you see two lattice copy attempts. What the simulation does, it does thousands of this, okay? And every time you do a number of lattice copy attempts that's equal to the number of lattice sites in your grid, that constitutes one MCS or one time unit in your simulation. MCS stands for Monte Carlo step, okay? And that's the unit of time in your simulations. Okay, so that is how it works. You do this many, many times, and as you do this, your cell start to behave, and that's exactly what you have seen in this movie, is many of those lattice sites copy attempts. You're not seeing all of them, you're seeing them every like 1,000 of them you see, and you see that the cell slowly change shape, but what's happening is a series of these lattice site copy attempts. Does it make sense? I hope it does. Okay. Okay. There's a good question from Ria. Okay. Yes, you can play with those properties of what the cell should behave, right? And this all have an impact on how do you accept or reject the less competences. So they has to have different behaviors. So as you go over the course, you will be able to see how all the things you can do, all the things you can add as property of the cells, and then you can make your cell behave exactly the way you want it to behave according to the model system that you're interested. Okay. And you can have cells with nucleus inside, with membrane, with another organelle inside. Okay, there are a lot of things that you can do. Okay, so in these examples, I just use the property of this I want to grow or shrink because it's very easy to understand. Okay, but you can specify many different behaviors to the cell properties. So the cells and any object in CompuServe3D can have many types of behaviors and properties. They can have a volume, a surface area, a length. They can have some kind of aspect ratio. Maybe your cells want to be elongated. They have interactions with the neighbors. So two cell types would like to be together. So other two cell types do not like to be together. They want to move away. They have directed movement. They can chemotext toward a gradient. They chemotext away from a gradient. They can pull other cells. There are many things that you can do. And all of these behaviors that you want to add to your cells in your CompuServe3D model is done by different energy terms or in what's called the total energy or the cost function of your system, okay? And the way that it behaves, the way to decide whether this pixel copy attempt is desirable or not is you just see how your effective energy of your cells and your system change with that copy attempt, and that will define if it's minimizing, then you're driving the cells, into the target behavior, you're increasing, you're moving the cells away from target behavior. Okay, we'll go into each one of those energy terms as we go along in the course. So when we accept or when we reject the copy attempt, so let's now dive into exactly how this works. Okay, so here the cell is here and have a current kind of energy, initial energy, right? And then you do this copy attempt, you select this pixel site, then select this neighboring pixel site and try to move this over here. And if you do this copy attempt, then your cell will have a different energy, HF, final energy, after this, okay? So what we do is that we make the selection, we know what the energy of the system is before you do anything, and then we calculate, okay, what will be the energy of the system if I do this copy flip, this copy attempt, okay? So that's going to be our HF. And by comparing those, we will have how much the energy will change between before the copy and after the copy, okay? And yet it's still a hypothetical copy, we have not made the change yet, okay? But we now know what's going to be the difference in energies, okay? So, and this difference in energy will be either higher or lower, right? Or maybe the same if there was inert, okay? So if this drives your energy lower, okay, so, and usually you write those energy terms in a way that they are zero when the cell is doing exactly what you want it to do, and it's higher when the cell is moving away from what it's want to do. Okay, that's how you write those energy terms. So if you are minimizing, then you move the cell towards its target behavior or target property. So if your delta H is negative, then that's a good sign. The cell is behaving exactly how you want it to behave. So you always accept that that pixel flip. So if you're driving your energy from, if your difference energy is negative, right? So you minimize the energy by the pixel copy, then you always accept it. And now you have a cell that's have this different shape here. However, if you are moving your cell to the, if this pixel copy actually moves away from starting behavior, then that's usually reflected by a positive changing energy by the pixel flip. And now you may or may not accept this pixel copy, and that will depend exactly on how far away you're moving the cell from its target behavior or property, or how big is going to be this energy increase if you do that pixel copy, right? So if you just slightly increase the energy, right, you're more likely to accept this because your probability of acceptance of this site copy peaks is very close to 1 or close to 100%. So let's say you accept on 90% of the times, but you reject 10% of them. But if you're driving very far away yourself from its target behavior or target property, then you're more likely to reject that copy attempt. You don't want the cell to change its shape in that way, so you're more likely to reject. So the way we do this, we just calculate the probability of accepting a pixel copy that drives the cell away from its behavior by using exponential, right? So it goes like exponential to the minus of the energy divided by some parameter t, which is, you maybe hear about parameter referred as the temperature of a system, but you should maybe, is better to understand it if you think of it as what's going to be the noise level of your system, or you can also interpret this parameter t as the membrane fluctuation level of your system. So the higher you make this temperature, you're more likely to accept any pixel flip. If you make this temperature 0, right, then you only accept pixel flips that are minimize the energy of your system. Okay, and there are cases when you want to make this kind of noise level higher or lower, right? If it's lower, your system will quickly go to its target behavior, right? But you may get stuck in an intermediate state. If you make this noise level too high, anything can happen and you may disintegrate your cells. So you have to find the correct level. So you allow the cells to explore a little bit the space around and eventually find their way into the correct behavior. Okay. So the way you model the energy terms that goes into this this effective energy is by using constraints. Okay, so in the case for the volume, you say that the cells has a target volume, right? And you compare that with the current volume of the cell, which is going to be just the number of lattice size that you occupy. And you look at the difference, you square, so you want it to be, you penalize deviation either to more area or less area. And then there's usually another term here, which are those lambda terms that gonna tell how strong you want your system to stay within its target area or deviate from its target area. Okay, so a stronger lambda will make the cells they will not like to deviate much. If you make this lambda parameter smaller, then the cells are more malleable. You can change its volume more easily. Okay, and then the same thing that you do with the surface, you can also add to the volume, you can also add to the surface of the cell or the perimeter of the cell if you're a two-dimensional simulation. You can also do the same as for the target length. And basically, most of the properties that you add to the cell, they have this kind of quadratic-like, they look like a hook and spring shape where you deviate, you penalize deviation in either direction of what should be the target property that your cell should have. Okay, so the further away you are, the more costly it is. So what is this pixel attempts are doing is allow your cell to fluctuate around target volume, and going up in either direction is penalized, but still allowed a little bit. But going down is always favored and you accept it. If you set your lambda to 0, you're basically turning off that behavior, right? So if you set your lambda surface to be some value, but set the lambda volume to be 0, then the cell does not care if it's very big or small. All it cares is about having a target surface area. So thanks for the question. When you set the lambda 0, you basically turn it off. Avishek, you have a question.
Yeah. So, what is the length L target?
That's for that's when you want to add the aspect ratio to the cell. Okay, so the way this works is you compute the moment of inertia of the cell, okay, and you find the major and the minor axis, okay, and then you match the major axis length to this target length, okay. So you won't say that the cell should have a major axis of length of I don't know 20 pixels, and this one right here has 15 pixels more or less, okay? Then he wants to change its shape in order so that this length here matches this target length here in this direction, okay? So it is another way that you can specify the shape of the cell to be, so you want to have elongated cells. That's one way to do it. Make sense?
Okay.
But
And suppose there is no growth and I just apply a force and there is like a translation, linear translation of the entire cell without any deformation. So the total energy will be 0.
I'm not sure I understand your question.
So for example, if I have a cell, right?
Yes.
And suppose I just apply a force and just make it move without
Yes.
changing its shape or anything else. So will the total energy be 0?
In the ComCell 3D, in the Cellular Potts Model, the cells always have to change their shape in order to move, right? So they have to transiently increase their volume and decrease their volume, right? So that's why you do not want to set this to a very high value. But as it moves in a way that, okay, it may just transiently change its shape a little bit and its volume a little bit, but you keep very close to the target volume and the target surface and the target length, even though you add a external force. There are going to be a whole section about adding forces to the cells, okay? And then you see how it goes.
Okay.
Does it
So
answer your
because
question?
of because of the growth, it's like the total age is not zero because of the growth, or like the negative growth assuming age, basically. Is
Yeah,
that
I'm not sure that I understand your question, but don't worry very much about that now, because we will teach each one of those, how to add each one of those behaviors as we go along in the course,
okay? Thank
right?
you.
Sure, no problem. All right, so the way it behaves, okay, you can add this volume term, and then you can also have surface term, a length term, okay? And they all contribute to your total energy, okay? And the way it works is that, okay, let's say the cell has a kind of volume V, small, low capsule, that is lower than this ideal volume, okay? And then you go from a state that if like this to this, so you're is a pixel copy attempt that increase the volume of the cell, okay? So this uh, is a case where if you look at your volume constraint, because the cell has a volume that's far away from its target, uh, it's lower than it is, so it has some kind of very high energy. And this site copy attempt, you're actually going to drive the energy of your cell from here to here. So you're going to lower the energy of the cell and it's driving towards its target volume. So this is a pixel site copy attempt that you accept because you are driving the cell into its target behavior, which in this case is to have this particular volume here. So this site copy decrease the energy, okay, because you're moving this closer to this, so this difference here is getting smaller, okay, so the delta energy of this site copy is negative, so you are driving towards its target behavior, so you are in a region that you always accept the pixel copy attempt, okay? So that's why you accept, and the cell now is a little bit bigger than what it was before. But suppose that the current volume of the cell, same copy attempt, but you set the target volume to be lower. So the current volume is actually higher than the ideal volume. So your current volume here, and if you accept this fixed copy attempt, the new volume will be here, it's going to even be higher. So actually you're already away from the ideal energy, and the copy attempt that you are trying to do, you actually make the cell bigger than even bigger than what it's supposed to be. So this is a case where if you do this copy attempt, you'll be increasing the energy of the cell, okay, moving it away from its target behavior. So that's a case where you are in a regime where the changing energy is positive, and you may or may not accept that copy attempt. So, well, during your simulation, the cells will be having forces from all the neighbors. A lot of things will be going on. The cell may find itself transiently in a situation where it has a higher volume than it should be. Okay, maybe the cell is being pulled on the outside, so it's being stretched. Okay, so and this force continues. So you're moving the cell to have a mean stretch, so having a higher and higher volume, right? And then the next copy attempt that you do, always, you remember, you always do this randomly. So sometimes you're going to decrease, sometimes you're going to increase, okay? So the cells are already being stretched, and then you have a copy attempt that's going to be stretched even more or make it even bigger. So that's something, the more you stretch, the more less like you are, the cell are going to accept further changes that are going to make it stretch even further, right? So going to resist more to the being stretched more to make it have a higher volume, okay? So this is a case that you likely are quickly driving, going down here, and you're more likely to reject that side copy attempt, okay? You can also be the reverse. Maybe the cell is being compressed by all the cells around it. They are growing and adding force to the cell, so the cell is being compressed, right? So that would be the situation before, that the cell has a volume that's very far away from the ideal volume. The cell wants to be this big, but because of the pressure of the neighboring cells is actually being squeezed. So in this case, a copy attempt that actually drives the cell to increase its volume is going to be highly favored. Okay. It can also be osmotic pressure or the process of cell growth. The cell is just growing and it wants to increase its volume, right? Okay, so in the case that the cell, let's say, is being stretched, and now you make a copy attempt that are going to increase the volume even further, right? So this, you're going to increase your energy of your cell even further than what it was before from the lattice site copy attempt. And then this is a reflects on a change in your energy that is positive. And so in this case here, you have a 40% likelihood that you're going to accept this lattice site copy attempt and a 60% chance that you're going to reject it. Okay, so you're more likely to reject than accept. And so you most likely reject that copy attempt. And then what happens next is that the cell shape is not changed. And then you proceed to the next random selection of a site to flip. Okay. Okay, so that is how it works. Okay. And then you can ask, okay, what exactly changed when you change the temperature of the system? So, or the noise level. So usually your simulation will have a fixed parameter T, which is going to be the temperature or the noise level of your system, okay? And this T variable, remember, is used to set the shape of this function here that's going to tell you exactly how much you're like to accept or reject a pixel flip, depending on how big is this change in energy that you have associated with that pixel site flip. Okay, so if for a given temperature, this will be the curve. Okay, so for this pixel flip here, let's say you are changing the energy by this amount. Okay, so we have a 40% likelihood they're going to accept this flip and a 6% likelihood they're going to reject it. And what CompuCell does is just generate a random number, and the random number has you compare with this number. If it's a random number between 0 and 1, of course. And if it's less than 0.4, it accepts. It is higher than 0.4, it rejects. Okay. So what happens when you change this T parameter or this temperature parameter is that you change the shape of this acceptance function. Okay, so if you make it a lower, then your explanation is more steep. Okay, and now for the same change in changing energy, you are more likely to reject. You have an 8% chance of rejecting the pixel flip, and the cell going to stay the original shape, and you have only 20% probability of accepting the lattice copy attempt, and now your cell has grown. Conversely, if you make your temperature higher, right, then this becomes flatter. And so what happens is now that for the same change in energy, you're now more likely to accept. You have a acceptance probability of 6%, and you only have a rejection probability of 4%. So the cell is more likely to deviate from its target properties and and to grow in this case. Answering very good questions you guys are put in the chat. Answering Kimberly question, yes, you can make this have different possibilities. And you usually do that by assigning different lambda volumes for different cell types. So some of them have a higher one, so they're more stiff, right? They have a higher bulk modulus, right? So it's harder to change their shape. And you can make some cells more compliance by having more soft. They have a lower bulk modulus, so you make this lambda value to be lower. And then for those cell types, it's easier to squish them or to stretch them. Okay. So you usually do this at the level of the individual parameters, and you keep the temperature the same for all the cells. There is a way to make the temperature different for different cell types, okay, but it's better if you do this at this level here, because then you have a better correspondence. You can interpret the parameters in a better way, in a more clear way. Does that answer your question, Kimberly?
Yeah.
Okay, great. Thank you. Very good questions. Okay. You're right, anticipating a lot of things. The way to convert between pixel sizes, you basically, that's a choice that you do right at the beginning. You choose, okay, do I want to represent my cell with 10 pixels, or do I want to represent my cell with 100 pixels? So that's going to your choice. Okay, how going to be the resolution of our system? Okay. But you cannot do that midway in the simulation. No. Okay, so, and again, as what I explained for the volume is also true for the surface, also true for the length, okay? And you may have combined experiments in a way that there is some kind of competition between either of those. Sometimes the same pixel flip, let's call flip, may be minimizing your volume, but maybe increasing your surface, okay? And then whatever you're going to accept or whatever this change is going to be reflecting an increase or decrease of energy, it will depend on the relative weights that you give to the volume, to the surface, to the length. So you may have properties of the cell that are slightly at odds with one another. And that's nice because then you can change one parameter and you can drive your cell to behave more in this way or more in that way. So that goes into the model design of our system, okay? So that's how it goes. So basically, that's everything that goes in a nutshell. You're basically adding properties of your cell by usually using this kind of constraints that have a quadratic term. So you have a very well-defined property of the cell, and that is this target property, and you can calculate what's the current value of this property, and you compare the two of them, and you search that any deviation for its target property or target behavior is penalized and reflects on a higher energy. And then what the comp cell does, right, it sums the contribution of the small energy difference for all those terms, sum all of them, and that's going to decide whether your pixel flip is positive or negative, and whether it's going to accept or reject, okay? Okay, so, and that's how you do, and then in every pixel copy attempt that you do, you're changing those energies in a different way, okay? You sum all of them, and that's gonna drive the behavioral cell. So although the lattice copy sites are done in a completely random way, their acceptance always reflect the behavior that you put into the cell, that you put in the model, so the cell will not move even though the lattice copy attempts are random, the movement, the end movement of the cell and the behavior of the cell is going to reflect whatever you put in these energy terms. Okay, so it's kind of is a guided, so it's a stochastic model. The cells usually move in the way that you design them to move, but in kind of random way. Okay, and you can decrease or increase this randomness by change that temperature behavior. Usually you don't want to make your temperature 0, because if you only accept things that move yourself towards your target behavior, you quickly freeze your system. but at the same time, if you make your temperature high or noise level super high, you're basically not seeing the weight of these functions here, and you're accepting everything, and your cell gonna evaporate, and you get just you get nothing on your system. Right? You don't have a model anymore. You have just kind of noise and randomness. So this is how it works, and of course, then you have to make a very good choice about how to change those things in a way that your cells actually behave in a sensible way. So what I like to do next is ask you guys to see those two models here. So this is a simulation where the cell is here, okay, and it wants to move in this direction, okay. The way that's done, the cells have those energy terms, and there's an external force that makes the cells move from here to here and from here to here, right? But I made different choice for my temperature and the weight of my gradient one versus the other. And I want you guys to look at those two simulations, which had the same output, are done in the same simulation model, okay, have the same constraints on them, and tell me which one looks more realistic and which one looks less realistic, okay? So I'm gonna play the one on the left and now the one on the right. So they're looping, okay? They're both two cells, okay? They're both moving the way that I want them to move. But tell me which one you think is a good model output and which one you think is a bad model output. Type on the chat. Is it on the left or the one on the right?
I think on the left.
Left, why?
Because the structure of the cell seems to be kind of like preserved, while that on the right is kind of like has a much bigger distortion.
Yes.
I might be wrong.
Okay. So does anyone think that the one on the right is the better one? I don't think so, right? Everybody thinks is the one on the left, right? Because usually when the cells move, they keep their integrity, right? They do not break into many parts. And certainly they do not break into 2 or 3 or 4 and then join again, okay? Sometimes when the cells move, they do actually leave a little piece behind that are quickly lost. So that might be a desirable output of your model. But for most cases, what you want to do is you want to have your model behave as in the left one. So why I show this? Because as we go along in the course, I don't want you guys to only look at metrics, okay, and numbers out of simulation. I want you guys to develop the sense of looking at the simulation output and know if the cells are behaving in a sensible way or not, okay? And I want you learn how to actually change the parameters in mid-weight simulation, use the player, okay? And the more you play with those parameters and more you look at what the cells are behaving with your eye first, the better you develop a sense of, okay, is this model behaving in a sensible way or not? Because your model may be given the correct metric output that you want, but sometimes when you look at simulation, you may be scared about what the cells are actually doing. So one objective of this course is and for you guys to take this course live and interact with us is that we can give you point you okay to develop the sense of if your simulation is look good or not. Okay. Yes, I can give the simulation to you. I think I still have it and I can make this available. Okay. But before I do so this is how a single cell behave. Okay. Let's look at another example. So this the level single cell. Let's look at this one. Oh, sorry. So here now you're looking at assembled cells. Okay. And what's going on is that you have a cell type blue and a cell type green, and they have different contact energies, and they want to sort out. Now, again, I want to look at those two models done in CompCell, use the same set of parameters, right? So, sorry, same set of energy terms, but they're going to behave very different from one another, and want to tell me which one you think behave the best, the one on the left or the one on the right, and why. Okay, and this is a one that it might be not very clear what will be the best one. So this and this. Now tell me on the chat, what do you think is more realistic? Okay, all of you said left. Okay. Okay, why is the left? Now type in the chat why the left. One of you already said something, but I want to hear your own thoughts. So the cell bonds on the right are too convoluted. Okay, what else is not quite right in the right? So thanks for your answers. So again, it's a situation where the output of the models, right? So if you look at the very last frame, is very similar, right? Actually, if you look at like this, they both kind of have an intermediate state where the initial state is the same. The middle state also is the same. If you just look at if this is a cell sorting and engulfment problem, the green is engulfing the blue, right? Even have this kind of same intermediate state. We have some kind of um, incursion here that is slowly going out. But if you just look at the metric without taking into account the shape of the cells, you even may say that this one is better because this has a lower boundary between blue and green than this one has. Yet, when you look at simulation, you see, okay, which one you believe? I believe the one on the left, because the one on the left looks more like real cells than the one on the right. Okay, so you always have to look at your cells and see that their cells look more like the ones on the left than the ones on the right. And one of the reasons that I want to stress this is that I sometimes I do see published models, okay, where the cells look very much like the ones on the right, or the movement of the cell when I look at when I look at still pictures, the cell look right, but when I look at the movies, the cells look the one behind that they're moving, but they're completely disassembling themselves in the process, right? So you have
Depends on the can also depend on the what is happening to the cell. If it is going through necrosis,
yeah, okay. In that case, then you expect the cell to kind of have very weird shapes transiently, but then eventually disappear, right? If it's going to necrosis or paroptosis, okay, you expect cell shape to be very weird, but for a short period of time, and the cell is not interacting with other cells. So in this case here, well, well, but yeah, it's a good point. Sometimes the cells do have weird shapes, and you want that to appear in our model, okay? But I'll say that in this case, a little bit less clear, wherever you cannot immediately say that the cells have on the right, have the wrong behavior or the wrong shapes, because if you're talking about epithelial cells, they do kind of look like more squarish, more cuboidal than these ones, okay? And if you're talking about mesenchymal cells, they do this shape here. Actually, for this cell, for example, that's very rough edges and kind of making a curve, is a better shape for a mesenchymal cell than these ones here, okay? So of course, if you look at the cells in the middle, it's a complete mess, so it's likely wrong here. But some cell like this one, I hope you are seeing my, can you see my mouse? Okay, so this cell here in particular that I'm highlighting is actually a very good cell shape for a mesenchymal cell on a substrate, okay? So that's by itself not really wrong, but you see this mess here, this is kind of wrong, not good. And this by itself is not really a, okay, this is completely squarish, so that's bad. But if you look at this cell here, that's not by itself a bad cell shape if you're modeling a highly cuboidal cells that exist in epithelium, okay? So maybe if those cells green are supposed to be epithelial, right? In a cuboidal type, maybe this is more wrong than this, okay? So you always have to not look at the cells, okay? So does your cell shapes look healthy in simulation? You also have to ask, okay, besides this, are they matching the shape that they have in real life, okay? Because sometimes a very well-rounded cell is a cell that only exists in the cell biology textbook. Is not a cell that should have the shape in the particular system that you are modeling, okay? But hopefully we're gonna give you guys all the tools for that you can actually play to make the cells a little more round, a little more mesenchymal, okay, a little more cuboidal, okay, and then you can actually choose wherever your maybe not have these behaviors on here on the left, but have something intermediate between what you see here on the left and what you see here on the right, okay? So so in the next module, our objective is to teach you all those constraints that you can add in our energy terms and how they affect the cell shape, the cell behavior, the cell movement, and interaction with other cells, okay? What each constraint and plugin that exists in cell can do and what it cannot do, how you can combine plugins to get an desired output that they alone will not give you, and how, as you do the exercises and see the examples, develop the feeling of how to choose the parameters so that your simulations look right, not according to some textbook, but look right according to the specific cellular or developmental or disease process that you are trying to replicate and study using computer simulations, okay? So this is the end of my session here. Okay, if I'm open here to questions, do not know how much time we have left, but please take your time to fill the module questionnaire or send more detailed feedback to Hayden using his email. And I thank you for your attention, and I'm very happy to take any questions you guys have or comments to here. Okay, so there was a question from Kimberly about what about killing a cell? Perforin release out of autophagy made disintegration of structures appropriate for the model. Yes, that's what one of your colleagues said. Maybe you want a cell to have a translated very weird shape that eventually disappears. One way to kill the cells in CompCell3D is by setting the lambda volume of your cell to be 0. So let's say your cell wants to disappear. Sorry, I didn't say. Sorry, I misspoke. One way to kill a cell in your simulation is by setting its target volume to be 0. Okay, then the cell starts to shrink and eventually disappears. Okay, maybe only set the target volume of the cell to 0 will not be sufficient for it to shrink, disappear, because it may have competing forces. So one thing that you can do, you can set the target volume of the cell to be 0 and set the lambda volume to be very high, and that makes your cell disappear very quickly. So those parameters here is not something that is rigid and fixed, okay? You can set the initial value for them, and you can evolve them over time in your simulation, so you can make your target volume of the cell bigger over time, and that will induce the cell to grow, or you can make the target volume to get lower over time, so you make your cell shrink. And you can also change this parameter here to make your cell more stiff or less stiff. In the case of autophagy, one trick that you can do, because the cells shrink and also have weird shapes, you can set the target volume of the cell to be 0, right? So you're gonna shrink, but you can also set the target surface of the cell to be very high. So that is gonna shrink, but also fragment to many pieces, right? And that will be a way to represent this autophagy of the cell where the cell disintegrates. Does it make sense? So
yes, very cool.
Yeah, you can do this. Okay.
Yeah, very cool.
So another question from Kimberly: can CompuServe3D create neural cell shapes? Yeah, you can also use the trick. You keep the environment to be something, and then you make the target periphery or target surface to be very high, and then you have a lot of protrusion of the cell. To get it the narrow shape, you probably want to now use maybe the length constraint. So you, besides have a very rough shape, because you have a very long target surface compared to what is the target volume, you also make a target length. So the cell, besides this, you also want to be elongated in one direction. And that will not necessarily make you a neuron-like type, because you have roughness all the way, but you're getting closer to a neural cell shape, right? Because in a neuron, you want to be very elongated and smooth on one side and very rough on the other side. But one thing that you can do, and you're going to learn later in this course, is that you can make your cell with 2 compartments, okay? Okay, so instead of our cell made of just one kind of pixel size, only one compartment can have 2, and then the one compartment is going to be the trunk of the neuron. So it's going to be very elongated and smooth, and the other part of your cell, you make it very rough, okay? And that's going to have a shape that looks a little bit more like a neuron. So your cell have 2 compartments, the body, and then where you have your dendrites. Okay. So that's using the cell compartments. Okay, so that's something that you're gonna only touch on Saturday, okay? But you learn how to do this. Okay, so I guess what I just said answers the question from Rama. Can some parts of the cell be adjusted differently from the others? Yes. Then you build up your cell made of different compartments, and each compartment will have different properties, right? For example, if you want to do a epithelial cell, you maybe want to split your cell into a basal compartment and an apical compartment. You do have completely different properties. The apical compartment may pull the other apical compartment, so you have apical constriction, and the basal site will preferentially stick to another cell object in CompuCell, which represents the extracellular matrix, for example. So you can do this. So there are a lot of interesting discussions going on in the chat. That's great. Not even sure I can keep up with all of them. Okay. If you want to ask a question just by unmuting your phone, you can also do this if you want.
I have a question. So, is it possible to calculate the stresses on the strain developed in the cell due to this kind of like deformations? And if
Yeah.
that is possible, Is that related? Can that be related to the H total,
To the what?
to the total energy of the system?
Yes. Okay. There's a whole paper about that. Okay, that given the energy terms that you have in your computer 3D simulation or your Cellular Potts model, you can actually calculate the forces at the bounds of the cell, where the forces are, where the cell is more likely to grow, where the cell is more likely to shrink. This is the work by Lisa and Hans. I don't have the reference here, but yes, only for the shape of the cell, we can calculate this kind of the stresses on the cell. Okay, if in a more simple case, you can also only take the what is your target volume, your current volume of the cell, and the lambda volume, and from that you can calculate what's going to be the bulk pressure on your cell. This strain is usually, well, only the difference between, well, you have to define what strain you're talking about, the longitudinal strain, bulk strain, shear strain, right? But you calculate directly from the shape, okay? And what will be the ideal shape or the initial shape, and then you calculate the strain. And you can also, if you want to add things like longitudinal strain or Young models along one direction of your cell, you can also do that. You can, if you have a cell with different compartments, you can add spring links that connect each one of those compartments, and by and the springs in, well, you can change the stiffness of the spring, and that stiffness is going to be direct proportional to the longitudinal modulus or the longitudinal Young modulus of the cell. Okay, so you can do that directly in a more explicit way. So there are a lot of things that can be done. So I guess the short answer to your question is yes, you can calculate the stress and strains. You're gonna require a little bit more work from your time, from your side, but it can be done.
Thank you.
Yasmin, I just had a question here. Is there a way to incorporate gravitational forces into a model? Yes, there is. You can add them directly by just adding a constant force on all your cell compartments that pointing down, okay? So that's a way to add the gravitational forces. That may be very important in some situations where the viscosity surrounding your cell is not that high. Because usually that's is not important because your cells are in a highly viscous environment and gravity does not play a role, but you can add a constant force around your cells. And if you add, you certainly can add a constant force on all the objects on your simulation that pull them down or in specific direction. So in short, you can do that. Yes. Okay, could you add viscous drag forces? That's a little bit more complicated. I don't know if James wants to come in, but there is intrinsic viscosity, drag viscosity to the movement of our cells just because they are in lattice, okay? Adding more explicit than this, I do not know if anyone has attempted or if it can be done. James, do you have any idea about this?
There's a viscous drag module in CompuCell, although I don't know if it's working at the moment. People don't use it very often, but you can do it. It's computationally very expensive, though.
But can we simply add some kind of like boundary conditions on the boundary cells or the lattices? Like, can we incorporate that kind of like modeling?
I guess depending on what you mean by boundary conditions, you can certainly add contact energy terms. That's something that we'll be talking about later. So the cell can have a different contact interaction with cells of different types of the medium. and that can be favorable or unfavorable, and that can induce a maximization of that boundary or minimization of the boundary. Okay, so that can be used to model the intrinsic contractility of the cell, or can be used to model the adhesion contact between cell of different types, or even the movement of the cell towards a specific point in space, and also has an effect on the cell shape. So you can there's a contact energy term that I'm going to be talk about later, that cells of different types have between themselves. So we have a matrix of contact energy terms between cells of different types. I don't know if that direct answer your question, but you can modify the contact, the boundaries of the cell with some other object.
Okay. And I have one more question. So is the final configuration, so given an initial configuration, you have the V target, S target, L target, the small l, small s, and small v. And given all of those information, is the final configuration unique?
No,
I
usually no.
don't think it is.
Usually no, because well, that depends on what will be your initial condition and how many cells you have in a simulation, right? So, for example, on this one, sorry, okay, in a certain sense, the final configuration is we chose the contact energies, such that the cells will sort out, and for that particular combination of contact energies, blue, green, and blue-green, and both with the medium, it's a set that always favors the blues to be inside and the greens to be enveloping the blue. Okay, so for this particular case, we know what are the conditions, right? And we know that we model the energy terms within these constraints. That's going to be the final shape it will have. But that also depends on wherever you have the correct proportion of cells, right? If I have much more blue cells than green, then the engulfment may be just not possible. All right.
Okay.
Or you have many greens than blues, the blues will never find themselves. Two different blue colors will never find themselves. So it would be in a metastable state that in the intermediate state that never happens. Or the blue cell, you may have so many green cells compared to blue cells that the blue cells will never see each other. So it doesn't matter what you do with the energy, they spread out. Okay.
Okay.
So even though the energy will be the same, the initial conditions also dictate whatever you can achieve the final shape or not.
Okay.
But of course, this is in a very simple way, right? And where you're only playing with the contact energies. Okay. If you go to more complex simulation where the cells also have different volume targets, so the cells are growing and dividing, so everything is dynamic, and you have morphogens and gradients going in, then your system becomes so complicated that you basically have to do the simulation to know what's going to happen, right?
Yeah.
So some cases you know what the final state will be. That still depends on your initial conditions and the spatial initial conditions of your system. But in most cases, you do not really know. You know, okay, for this parameter, it should go in this way. For that parameter, should go in that way, right? But when you put them together, I don't really know what's going on. And that's exactly the type of situation that you justify creating a model, okay? Because you want to know exactly one behavior is favorable than the other set of behaviors, or whatever third behavior that you do not anticipate will happen. So you have some emergence happening in your simulation, which will be even more exciting. Okay.
Okay. Okay. Well, thank you.
Sure. Okay, if there is not, I thank you guys for your attention, and I'll be back with you guys to teach other modules on Saturday and Sunday. Okay.
Julio, I have another question.
Oh yes, please go.
Are energetics influenced by cell-to-cell communication or even just chemicals in the surrounding or like chemotaxis? Is there, I'm thinking of like active transport, passive transport. This is kind of a weird question. I don't
Well-
know if the energy of a cell is lowered or raised at all from processes of neighboring cells within communication. That might have been answered before with endocytosis or something.
Okay, so endocytosis is usually a very small process in terms of scale that I'm not sure we can be explicitly represented. Maybe it can be done in an indirect way, but we can simulate diffusion of morphogens in CompCell 3D and make the cells respond to the morphogens by usually the way the cells respond is by chemotaxis. They move towards the morphogen or away from the morphogen, but it can also respond to the morphogens by using it as a trigger to change some cell behavior, change the cell type, or change some internal state of the cell. Okay, so you can add morphogens to your simulations. You can also, so if you talk about internal state of the cell, right? Can be its metabolic state or gene regulatory state of the cell. That is usually done with OD models, ordinary differential equations, or the SBML models. That's something that you can also add to CompCell cells. You can develop a model and save it as SBML format, or directly write the equation of your metabolic state or energetic state or gene regulatory state and plug inside each one of the cells and have one of the inputs of this internal network model inside your cell to be the morphogen or to be the contact that the cell has with neighbors.
Yeah, okay.
So this is something that can be done and will be taught in this course, okay? So it's not the Cellular Potts Model, the CompCell 3D. The basic is what I shown here, is represent the cells as pixels and move those pixels according to the energy. But on top of that, you can add those OD models inside each cell and also have morphogens on top and have all the three layers communicate with one another.
Yeah. Okay, yeah. That makes a lot of sense.
Yeah, you have all this flexibility.
Mm-hmm. Cool. Yeah, because it's movement, it's also growth or contraction.
Yes.
Yeah. And it's a before state and an after state. So the metabolic state could be influenced by the surrounding environment.
Yeah. So each
Yeah.
cell have the same kind of metabolic network, but at different states depending on their local environment. You can do
Yeah.
that.
Mm-hmm. Cool. Thank you.
Sure. All right, I think we have to stop here so we can have a little break before the next session. Again, I thank you all for your attention, and I'm going to be seeing you again on Saturday and Sunday. Okay, bye-bye.